but bear with that. But the idea that I will present here will be very pertinent to the group of people and the participants, because you all come from or loosely connected with the computational biology, as I understand. So the idea is how we can integrate a lot of atlases. So when I did PhD, there was no atlas. When I'm done with PhD and now I'm a faculty, I see every other day or every other month a new atlas is coming, and those are magnanimous, right? And it can actually accelerate our biology research and getting insights from the data a big way. So today we are still working. We don't even have an engine, right? That's how the biological research is happening today. We're working in silos. And I think we can improve that. It's not a very difficult problem to solve. It's just a mission that we'll have to take up. So to start from the beginning, there are certain conditions that you acquire through genetics from your parents. And for example, you cannot probably read it because of this table. Eye color, hair color, Parkinson's, we had a lot of talks on that. We do Jiva studies to find out genetic phenotype associations. And we have certain SNPs that we catch as we go on in our life, and like cancer is one, and type 2 diabetes could be another, and there are many others. I don't know the entirety. So what has happened is, over the past few years, the technologies have become much better. So we have a lot of omics that we can perform at a very low cost today. In fact, all the Indian good institutions have omics facilities of their own. So it's not a difficult challenge. And there's a lot of data that is already out there. We can top it up with our own demographic data, as Ishan spoke of. But there's already plenty of data. But this data are not managed well. And it's a trivial task. So the grand challenge I'm talking about is a trivial challenge. So multi-omics data are basically looking at your cells and samples in different ways, right, from different perspectives. And those are all very useful perspectives. But what is not happening is aggregating this data, integrating this data, and learning through the integrations, right? So let me give you a couple of examples of how these landscapes and atlases look like. So this is the Human Cell Atlas, the first publication that came out from a Chinese group. It's about 0.7 million cells of human body from across 50 different tissues, and that's a huge data, right? We could not, maybe at that point of time, NCBI didn't have that much of bulk RNA sequencing samples, so that's a single study. Now you can imagine, as Ishan greatly spoke of, there are a lot of such studies are coming up, so we have huge, plenty of single cell data. Then you have the GTEx consortium, which came up with this genotype. They came up with, initially they came up with the expression data, which is huge in number, normal baseline expression of different tissues, which is 17,000 plus. And the genotype data also they are now augmenting. They are doing a lot of analysis proactively, genotype-phenotype associations, and those are excellent. So these are actually matched data, that is what I'm given to understand. So you can do this EQTL analysis and all that. One of the speakers yesterday talked about it. All right, so that is another great example of a consortium scale and community scale research and data creation for the public good. The other one is, if I look at the GWAS catalog today, it will have about 6,000 plus studies and 500,000 plus SNPs. So billions, hundreds of billions of dollars are invested in getting these results. So this is a great piece of learning that we have got. And these span across maybe 3,000, 4,000 different traits, right, and the conditions. GenomeMad, another excellent initiative, which basically is a bioinformatic initiative. Again, this is something that we should think about as Indian students and faculties, because we don't have the luxury of a lot of things, because our problems and priorities are very different from Western problems and priorities. We are not lagging. So that I just wanted to rectify. We are not lagging in anything. Our priorities are different, and now it has shifted to something else, and we are doing pretty good. Okay, so don't think yourself as lesser. Now, GenomeMad is having currently, I guess, 1,50,000 or something, to the tune of 1.5 lakhs genomic data integrated from across different studies, and it shows you the diversity of the SNPs and the rare SNPs and so on and so forth. A couple of days back, there was a paper in Nature, Main Nature, which published 1,000 cancer tumor single-cell RNA sequencing data. So I just said, wow, right? It's a single nucleus RNA sequencing data from 1,000 patients' tumors. So this kind of data are all available. Now, what I'm pitching for in this particular, in front of this crowd, is basically correcting them, when I say correct, that means, you'll have to remove some batch effects, right? Even if you don't do that, it is okay. There's a second level problem. Don't worry about batch effect removal. We know how to run algorithms. We know how to do mathematics. Those things are fine. But if you do it, that is even better. So integration is the first level of problem. The second is warehousing them properly. So right now, the way we manage this such deluge of data is very imperfect and suboptimal. So we need to properly warehouse this using the warehousing principles. This is basically software engineering. So far, I have just been talking about software engineering. And this grand challenge is all about software engineering, in which we lead the world. The third one is something that enables this bespoke queries. You can think of as a business intelligence tool like Cognos or Power BI sort of a thing, or API calls that you can make. So you should be able to query these integrated databases to get your bespoke data that you can use for your specific scientific use case. And if this happens, this is commercializable as well. We can sell it to the world. Like UCO Biobank is not giving it for free to people, by the way, right? So they are charging for it a good $10,000, $12,000 per year or something like that. What I'm also provoking for is basically the hybrid cloud kind of a model, which is cloud is not going to sustain at all. It's not affordable, first of all. Academia cannot afford it. And also, there's a big hassle to maintain your own big data centers, not that every institution can afford it, right? So hybrid cloud is going to emerge as a better solution, perhaps, where you have some private servers and you have some public servers and you have the securities, blockchain, whatnot. I'm not an expert of that, but I think this is where we'll have to head towards. I'll give you some fictitious examples which I cooked using ChatGPT. Like, had you have this kind of a integrative cohort of atlases, then what kind of queries you could have run. So these are some examples which may not make sense completely, like select variant, phenotype, gene expression from GWAS table and join EQTL on GWAS variant equal to equal. So you are kind of doing a lot of join operations, and this is exactly what we do. And we struggle also. The gene names don't match. You put it on Excel, it will give something else, right? It'll change flip the gene names, it will something else. So this kind of queries we can run. The second query is something like population variant phenotype from the GWAS data, population in the names of the populations, and so on and so forth. It makes your life so easy, right? So you can slice and dice the data for your specific scientific questions, and this is pretty much software engineering at this moment. The downstream good things, the mathematics, computing, these are not the biggest challenges. We know how to do it, right? But getting to here is the biggest challenge. With that, I'll stop, and I'll take a couple of questions if time permits.
One question.
Yeah. Maybe, and also the classical problem with NCBI is that they are harboring raw data. So we need fairly processed data sets to make it affordable and viable. Otherwise, it will not work. Otherwise, we'll waste a lot of dollars. Okay? All right. So I think I'll move.
Thank you. Next we have Dr. Ramesh.
Slides, but I did go over the some problems in my talk yesterday. So, but I'm going to go over two of those. One of them is a smaller achievable data available problem that can still make a big impact. The other is along the lines of maybe what Ishan presented, a larger scale thing that will need considerable amount of effort, but can make a big impact, and that India can potentially lead the world in. So the first one, as I went over yesterday, is in this area of rare diseases. So we do a fair amount of diagnostic testing, which means a rare disease patient comes in. What does that mean? They have typically their disease is caused by a mutation in the DNA, 1 or 2 mutations. To find those mutations, you have to sequence. That process has its complexities, as I went through yesterday. But the net of it is that using the best methods today, whether you do whole genome sequencing, whole exome, whatever, about 50, maybe 60, if you push it, % of the people get a diagnosis. The remaining 40 to 50% don't. They just pay for the test. We just run them back saying, here I found nothing. Right? So that's the situation today. That's the problem. Now, in many of these cases, the fact that it's a genetic issue is pretty clear. I mean, it's happening right at your birth or very early in life. It's sometimes there's a family history, a strong family history as well. So I don't think there's a doubt on the fact that there's a genetic basis to this, but we are not able to find it today, right? About 40 to 50% of the cases. So, as I said, we have thousands of blood samples archived where of undiagnosed cases. They came in, we couldn't solve them, and they're hopefully there in our biobank. So the samples are there. Whatever sequencing we ran is there. On top of that, we have to do a lot more perhaps to figure out where is this remaining 40%. And if we can dissect that 40% and get to whatever, even 10% improvement, that's like, probably 100,000 people are getting sequenced, and another 10% improvement on that can be 10,000 more people diagnosed perhaps, right? So it can make a fair amount of impact in the short term. So the question is, how do we go about doing something like this? What are the investigations we might do? Because genome sequencing has not worked. Maybe some fraction of it will, going to whole genome sequencing might get you there. You might have to do something else, maybe look at RNA sequencing to verify unknown things that you found in the genome. What else can you do? I mean, that's really the open question, and what can we do to solve this problem? So I'm calling it a grand challenge because rare disease burden in India is very large. If we solve the problem, India is unique in the sense that you will in no other country will you find that sort of clinical population of rare disease patients as you will find in India. There is data available in India. I mean, we have samples available, etc. Impact is readily there. So that's problem number one I would put out. And if groups of people can come together and we can figure out what to do with this problem, it's a real applied problem that we can try to solve. The second problem is a more futuristic, longer scale problem, and that has to do with cell-free DNA. So just to remind you of what that means is, of course, we have the DNA that we are born with, and that in some theoretical sense doesn't change through our lives. But then there are mutations that accumulate in certain cells. Some of those mutations lead to diseases. There are other sorts of non-mutational changes that accumulate, epigenomic changes. And over a period of time, your aging process has a strong mutational plus epigenomic feature that happens in different parts of the body in different ways. And eventually there's a disease state that one ends up in. And it's very hard to detect that disease state early enough. And it's happening in certain cells in your body, and out of the trillion cells, if there are, 100,000 cells somewhere in your liver or somewhere that are going bad, it's very hard to figure out what's happening until you get to a certain state where it becomes clinically apparent. Now, the one window that we have that we are realizing over the last 5 years or so is that you have cell-free DNA that comes from all different parts of all different cells, where the cells go through apoptosis or one of the other forms of cell death, and their DNA gets chewed up by various enzymes, and it presents in the blood as free-floating fragments of certain distinct size distributions depending on what enzymes have cut the DNA. Now, it's easy enough to get hold of that DNA, and it's now possible over the last 3 or 4 years that a number of companies, groups have published that you can measure mutations in that, you can measure methylation status in that, and most importantly, from a cancer perspective, you can also detect a number of cancer cases well before they become clinically apparent with a relatively high specificity rate. You can be in your 90s, perhaps even in your late 90s in specificity rate. So now that we have evidence in cancer, there are so many other diseases, chronic diseases that we suffer from, that we don't detect early enough. Can we create an atlas of cell-free DNA coming from a large number of individuals, some normal, some so presumably, since we want to detect early, we don't want people who are already down the disease spectrum, but perhaps we need to longitudinally create this for a large cohort. Let's say take 100,000 cohort, follow them up for 10 years, and many people will translate into many sorts of diseases over that process, and then you'll have cell-free DNA archives over 10 years. And then what can one discover from this on various sorts of diseases that can lead to early detection? Again, why is this relevant to India? We've got our own set of chronic diseases, we've got large patient burdens, um, early detection, no doubt can help. Many of these are lifestyle, changing lifestyle can postpone, prevent, etc. So, and this is something that the West is just about also waking up to, unlike tumors where there's large atlases available. Single cell is catching on very rapidly. There the atlas size of course is massive. Here also we will need a large enough atlas size, but you can decide to focus on certain diseases or focus on just taking 100,000 population, following it up for 10 years, and you'll get a sampling of a large number of chronic diseases that are prevalent in India, that's sure. Right? So there are plenty of both assay design problems and signal extraction problems from the data. When you look at cell-free DNA, the signal is very weak. You've got to design your assay to be able to maintain good control on variants so that you can detect the signal. So you'll have enough data, interesting data engineering challenges as well as we solve this. So those are my two broad areas that I'd like people to think about.
Second question also solve the first question in your case?
No. First question, would solving the second? No, it will not, because for the first question we already have the patients in front of us. We've got blood samples from them sitting in our bank. We just need time, effort and funding to explore from all possible angles what can we get from the blood of these patients. We can perhaps go back to these patients and get other tissues as well. In a fraction of those we can do that. So the samples are already there in some sense. Right? The second question, the samples have to be collected in large enough numbers so that we can mine the results. That's why I mentioned the first one, because it's low hanging. It can be off the ground right away because the samples are there.
And the question is, supposedly we succeed in doing the
second one,
would it?
I doubt very much. I think we'll have to, in the first one we have to dig deep in looking at it from multiple angles in terms of what signals can we get out from the blood or from any new sample that we can collect that can help diagnose this. I think somewhere in the DNA it's there for sure, but what is that? What is it? We don't know. So that's the
quick follow-up question. So the first problem is quite challenging and interesting, but one comment I wanted to add is, if you also look at, let's say, RNA-seq for instance, it could be different assays. If you also look at RNA-seq of the patients you have already diagnosed and you know the genetic factors, so it's unfortunate for those patients that they have these perturbations, but in the context of understanding general gene-gene interactions, if you do an RNA-seq, then you know which genes are perturbed if you have certain genes which are deleted or perturbed according to your DNA assay, this 40% that you've already characterized. So then you do your RNA-seq on the other patients and you can look at the comparison of those signatures to see if there is any similarity between these rare diseases in terms of their blood RNA-seq. That may help us point.
You can. So that's one of the tools that will get used in this process. I'm assuming one would do some sort of blood RNA-seq if the DNA sequencing has given you some variants that you're not sure what they do, unknown significance. Maybe looking at the blood RNA-seq will help.
That's right, but not just for the uncharacterized patients, but also for the characterized patients that you have, so that you have some reference to relate to it.
See, this is the spectrum is wide. The people who come in with a wide variety of diseases and genes, and so just looking at some handful of 5 people is not going to give us anything, right? So
yeah,
it's we need to think through how to do this, how to pick. If we need to look at characterized patients, how to pick them, and certainly uncharacterized patients, how do we throw the kitchen sink on them so that we can figure out. And once we know where to look for the signal, maybe that can then be applied in a very canned way in practice, and that can give us another 10% or whatever. But we need to get to that point.
that's right. A polygenic risk scores could be used for selection of patients in both both the first
big
and
things.
second.
Polygenic risk scores, of course, are very, very complicated to deal with in the sense that you will have on an individual basis, you will have no way to verify whether this polygenic risk score is the cause
That's
of
right, not
anything
at
or
an
not.
individual level,
Well,
but at
everything
a
we're
top.
doing is individual. I mean, the first problem is only at an individual level. It is useless at a population level, right? It has to be at an individual level. Otherwise, you don't have any impact whatsoever. So,
Thank you,
all
Dr.
right.
I hate to be the guy who has to break the interesting conversations, but remember that we have a discussion session later where we can, all of audience can ask questions and we can have a discussion with everyone's participation.
We're going to switch. Yeah, we're going to switch from a applied problem to a more modeling problem, a basic science problem, and also more from data generation to analysis of existing data sets. So this is about going from model organism databases to computable models such as whole cell models or whole body models. Okay, let me just give you a few examples of those models. So what do I mean by digital computable models or these whole cell or whole body models? So this was a representative example from 2012 where Carr et al. Built this whole cell model. It involved actually 28 submodels. And all of these models are pertaining to a single cell, and you have the metabolome, the genome, the transcriptome, proteome, all these different multiomics layers that were modeled across different time as a single cell divided into two different cells, right? So this is a single cell organism that they modeled. And the process that went into it is phenomenal in the sense that they had to read 900 plus publications. So there were mainly two authors who were kind of sharing, I believe, the first authorship, and then there were other people from Carr's group. So they had to read through these 900 plus publications and learn or fix these 1900 parameters. Like I said, there are 28 processes I will show in the next slide that they have to build. So that you could get this model that could predict the effects of different perturbations. So there was talks about digital twins earlier. So this is not an exact digital twin in the sense that it is representative of the whole organism, but this is one way to step towards digital twins, right? And these are some examples of the different processes in that, like protein processing, ribosome assembly, RNA modification, and these are the 28 submodels that were built. Okay. So going from whole cells to whole body models, so there are these metabolic based models. So there are people who are in the crowd who are more well versed in metabolic analysis than me. I mostly focus on gene expression analysis. But in the metabolic models that I have built for multiple organs, so Thiel et al., Swagatika Sahu, who is currently with Vantage, and so they have built this Harvey-Harvetta model, so very creatively named, so Harvey and Harvetta for the female and male version of the model. And this has more than 30 plus organs that are being metabolically modeled. And they look at, again, different compartments, right? Not just these organs, but all the blood cells. So here it's a bit harder to read. There are these 20 plus organs, 6 sex organs, 6 blood cell types, and more than 10 biofluid compartments, urine, plasma. All of those are being modeled like there is a genome scale metabolic model called Recon3 that are being adapted to each of these organs, and then there are exchange reactions that are set up to set up the communication between the different tissues. So we have made some small steps towards this. So Nilesh presented a poster yesterday from our group on trying to integrate gene expression and metabolism, and already it's quite challenging. But we believe that constructing these whole body models, at least certain aspects of these models are within reach. But one of the bottlenecks is extracting these parameters and making decisions about hyperparameters in these models, reading a lot of literature, hundreds and hundreds of literature to infer and to make these decisions, right? And fortunately for us, there are a whole set of curators from these different, who specialize in different organisms. So SGD is the yeast genome database, and Flybase is the Drosophila melanogaster, so fruit fly related database on all the genes in there, the proteome content, the gene-gene interactions, the protein interactions. So all of that are available in some form of semi-structured fashion in these model organism databases, right? By semi-structured, I mean that there are certain free-form texts where they say these are all the major publications related to the fly, and then these are some annotations that they have. These are the genes mentioned in these publications, and these are the relations between genes and diseases or genes and pathways in these publications. And as well as certain large-scale experiments on protein-protein interaction, they just have a list of protein-protein pairs, right? And these are really the components that go into these whole cell or whole body models, and they are already present in some way in these particular model organism databases. So one example is the East Genome Database. Like I said, these are kind of the 187,000 publications that they have curated to some extent, right? So they have a whole army of curators who go through these publications, and then they look at all of these different things on the left hand side here. So the biological processes, the molecular functions, diseases, cellular components, phenotypes, and all of them, right? So these are extracted from these publications, and some of them go into structured databases like gene ontology that we use quite often. But other things are just in raw files in this particular resource, or through searchable interfaces in these resources. Okay. And I was going to go into that, but in the interest of time, I will just say that, just end with this slide. So can we go from these model organism databases like SGD and Flybase, which contains a mix of these unstructured and structured information, a whole wealth of information, decades and decades of effort by lots of curators, right, into these digital models such as whole cell models or whole body models like Harvey and Harvetta, where you don't have to go through each of the literature manually, but somehow use the information that is already out there in an automated or semi-automated fashion. So the reason I believe that it is a well-suited problem for India, or a well-suited challenge for India, is that we have, like I said, it is mostly dry lab based. We are not generating new data, new experimental data. We are looking at existing data that has been generated worldwide and collected in these model organism databases. And then we are trying to construct a computable model. In some sense, the whole of science is trying to look at the wealth of information that is generated across all the publications and condense it into a model, a very compact model that we can use for predicting perturbations to certain genes or certain proteins, and to understand what it will do without actually conducting the experiments, right? So it will have huge long-term benefits in terms of if we are able to achieve it. And the reason that I feel India is suitable for this is we have a whole lot of experience with modeling, a whole lot of experience with, we have the best computer scientists in the world, the best machine learning people, so there will be a lot of text mining involved in order to understand this, and we are at the time where the natural language processing is making huge leaps with tools such as ChatGPT and other things. So there are certain steps in which you can achieve it. So reading these publications, so there was a question question earlier, right? So that Rukmini was asked on, rather than going through publication, have you used ChatGPT to speed up some of the processes? So that would be the first step to start. Can you accelerate some of these literature searches? But what would be ideal is that I just say this is a model organism database, and then feed it into a particular system, and that will just give a draft version of a computable model, which can later be refined based on syncing up with other experimental data sets. Okay, and then so I'll just stop there and then see if there are any questions.
In some of these model organism databases, heterogeneity is an issue, right?
Yes.
So how do you, that's the challenge. How do you plan to handle that?
That's right, yeah. So heterogeneity is an issue, but there are certain components like protein-protein interactions, a list of protein-protein interactions, the gene tables, right? So where the genes are located in the chromosomes, the functions of the genes, the gene ontology. So certain aspects of these databases are shared across these different model organisms. So we should probably start there, right? And then go on to other kind of intricacies of each of these databases. Yeah, and another kind of strength that we have is this involves a lot of mathematical modeling. So we have good mathematicians, engineers, and so there is already a lot of work in all the institutes, different institutes we are coming up. You are coming from, where we have these whole body or whole cell like models, but more on certain aspects of, let's say, transcriptome and metabolome alone, or metabolome alone. So can we just pool together all this expertise of the people who are present in this room already to start working towards a universal model that combines all of these components?
Just a short idea. Instead of going top down, can we flip the problem? If a whole cell model is done, can we now build an organism by putting, let's say, 2 different cells from different organs, like build an organ from sort of grow the model instead of fitting the model?
So if I understand your question correctly, so this whole cell and whole body models are two different components. So currently it's not like the whole cell models are put together to get a whole body model. Whole body model is a totally different kind of formulation, a linear programming formulation, and whole cell model has a lot of molecular simulations, Gillespie simulations and so on. So they are not talking to each other currently. So that would be another challenge in itself, how to reconcile those two models. But your question actually suggested an idea to me, which is you can start with this whole cell model that is already out there, and then you have now the names of the parameters, and you need to just know what these parameters are for these other organisms, right? So which we can try to learn from these model organism databases. Yeah, that gives you a more tractable route to go after.
It's like to do genome annotation from one genome to another.
Yes, yes, you
Model
transfer.
annotation and then have to. There are things like model seed and other tools which do that automatically for metabolic.
I see. Yeah, yeah. So what I'm suggesting is then can we grow to kind of other types of data, but you do a transferring the annotations from one organism to another organism where the whole cell models is already available.
So the problem that I thought of is taking one scientific problem and using data methods and computational modeling and then try and solve the problem. So that was what I thought. And all the slides were made by my graduate student Deepshika Singh, must be somewhere here, who is actually working on some aspects of this. Okay, so this is about type 2 diabetes. Why type 2 diabetes? We are the world leaders of type 2 diabetes, unfortunately, in India. And one maybe for lifestyle reasons, two for genetic reasons, and three for environmental reasons. All three things I think making diabetes a real burden in India. The problem with studying diabetes is that genetics is important. Okay, and genetics itself is it's a single word, but then it's a very very heterogeneous, and mutations like in cancer, driver mutations here too will be making a big influence. Interactions among them, environmental interactions, could even be the air quality. We all know the problems associated with air quality, and what we breathe, the kind of food we eat, how much pesticide in it is there in it, and everything associated. I mean, you can list 100 things of that kind. All of that has an influence on whether we get diabetes or not. Of course, there is type 1, type 2, and monogenic diseases. Diabetes itself is not a single disease, it's a big spectrum, right? That's another thing to note. From early onset here to, I think, a large number of people are out there, type 2 diabetes, pre-diabetes, diabetes with obesity, without obesity, with fatty involvement, without that involvement, and so on and so forth. So this is a big spectrum, so making it very difficult to study. Conventionally, the way it's being studied is as a clinical, a case control study. You have a case, which is a diabetic case, and you take a control, usually not one sample, but you take a cohort of people, and then you get some differences. Again, only if it is statistically significant, we blindly believe that, and it depends on the cohort you've chosen actually. And then you take that and leave everything else out, whatever. So those people who do not fall into some median range of the variations that you are looking at are all left out. It works only for people who are in the median range, and that depends on the kind of people you have enrolled in the first place. So that's a big problem. So there are, I think, of course, you can do genomics, you can do transcriptomics, metabolomics, whatever, proteomics and so on, but that is a problem. And currently, people are trying to characterize molecular heterogeneity by looking at what pathways, how much involvement is there, each one referring to one gene here and one pathway, whether it's upregulated, downregulated, but there's no clear consensus on how to approach it. There are so many variables, both in the genotype as well as in the phenotype. I think really one of the challenge, what I thought of was relating this genotype to phenotype. I think that's where we haven't made that much progress in any discipline, I mean any area of disease basically, but diabetes may be one of the most interesting ones to start with basically. And again, people are looking at hierarchical clustering or hard clustering, really group people into these different colors based on the traits. And I think a new approach, well, this really fails miserably. is to, I think there are publications which talk about soft clustering where you put them in more than one group and so on, or to do various types of multi-trait regression and try and find some combination of these traits which works and doesn't work. But all of this is critically dependent on deep phenotyping, clinical phenotyping of a group of patients. I think that's absolutely essential. And of course, all the data that we can collect, such as all the omics that are necessary, plus biochemical modeling, how well can we actually put everything that we know about factors that are associated with diabetes, both from a causal level as well as from a consequence point of level, then characterize that. Because once you have that kind of deep molecular characterization, then what will be important is big data analytics, because it will become too much for one single model or one single angle to get something meaningful out of that. So we will have to address multiple approaches, and this is where I think we have an opportunity to do that now, because the translational outputs, many questions can be asked, to start with the subgrouping, which best patient stratification for what is the best treatment among the available options that are available would be one question. What is the prognosis? Are there comorbidities that are likely to come in this individual if ABC are perturbed as opposed to, say, DEF being perturbed, and so on. So this is, I think, some of these things that we can actually look at. I think that's the last slide. So this is the challenge. I think many of us can come together perhaps to collect very clean data involving clinicians, involving very strict protocols and deep characterization, and then learning from that is what I got. Thank you.
There was a study on a single person, Michael Schneider, I think is a PI in Stanford. He did this longitudinally for himself, where he was collecting all these parameters, and he was able to actually predict that after a viral infection, his blood sugar levels and insulin was off. So do you think that, and there are there's also one tech founder who is posting videos of himself measuring, aging company guy. So there are, it just takes too much resources to do deep phenotyping. But do you think, like, given that such data is available, we can identify what parts of this daisy chain of data collection are more important, and what granularity of data collection could be required? Because genomics is once, but everything else has much more and you can collect data every day like that tech founder, or do it smartly.
Do it longitudinally over a period of time. We may have to choose time points. We have to prioritize from practical consideration, maybe once in 6 months for 3 time points or something of the kind. But clinical phenotyping is easier than we think. Many people collect, all sorts of blood tests are done. It's just that they're not organized, and no one reads from it much more than a second's glance by a physician and then saying you're fine, because that's all time they have, really. So we need to combine all of that together, plus lifestyle. We need descriptors to start with. What do we mean by lifestyle? What is the food intake? How do you categorize it? Right? How much do they cook? Nobody ever cares for asking these kind of questions. And what is their amount of number of steps they have walked, or any such thing. So those are the things to start with. What should the data even look like? I think that is an important thing. It's much easier than you think. I think with apps and so on, you can do crowdsourcing and then get this out.
Thank you, Dr.
Thank you.
Next week,
So in keeping with my previous talk, where I've actually donned the hat of an AI person rather than a modeler, which I usually am in these meetings, I think there are a lot of problems. I'm not going to outline one single grand challenge, but rather a battery of problems that cut across many of the presentations that were made so far, and I've actually not seen as much AI as is typically seen in these conferences today in this big data and biology conference. But just to walk around the next couple of floors shows you so many posters on AI for healthcare, AI for medical data analysis, and so on. So what is responsible AI? And I think a lot of us, including myself, who's not been fully exposed to this field, we totally recognize the importance of data privacy and things like that. But responsible AI is a lot more than that. Starting with AI and safety. And this is both in terms of stability of your predictions. Right? So when you say that a person is being diagnosed with cancer, right, how stable is your prediction? And what are the performance guarantees that you can give? And the next step is explainability. Right? So can you actually make AI understandable? So the running joke we have is that somebody has cancer because the explanation for cancer is that there are 4 neurons in the doctor's head that fired when he or she looked at the X-ray or whatever. Right? That is an explanation, but you can see how laughable that is. And I think we are at a point where the AI has to make those steps towards making it more understandable, to using a vocabulary that humans can appreciate and actually understand, and more importantly, the clinicians can trust and make decisions. And obviously, this also brings into force the human-in-the-loop systems and so on. Data protection, privacy, I think that is something that all of us understand, but a lot more needs to happen in India as well for this. AI and society, again, lots of open questions here. Ethical decision making, then of course future of jobs, perception of AI and so on. But even fairness, right? We're talking about so many ethnic groups in India and so on. So do we have proper representation from them when it comes to any kind of data? Could be type 2 diabetes data, could be other kinds of data that we are generating. Do we have enough representation from all these groups to make our learning more robust? So there are like so many of these questions, and I think last I would say is reproducibility. It applies to every field, but all the more in the context of AI. You must have heard of FAIR principles in terms of findable, interoperable, reusable, accessible data and so on. But can we actually make the FAIR data fairer now with reproducibility brought into the mix? Just good luck trying to, I think there are so many young PhD students here, so I'm sure you would have wasted multiple months of your PhD trying to recreate somebody else's study, and be it a modeling study or in AI it's become even harder, right? So have you actually, are people saving the random seeds, right? Can you reproduce the cross-validation results of some other paper? All these are big challenges that I think we need to quickly wake up and address, and these cut across practically all of the discussion that has happened so far. And all the more important in the context of biological data analysis and medical informatics, and requires constant and close discussion between various stakeholders, and which is why, you know, in groups such as these, we could make these working groups that can come together and discuss these in the context of the challenges that are being proposed right here. And I'll just, you know, talk about our center for a second, because this is actually not a single institutional center, but a bunch of people trying to come together, including the industries and so on, to actually do these things, right? So all of you are welcome to become part of this initiative. So we have this new up and coming center on responsible AI, but the whole idea is that we need to tackle this battery of problems, whatever be the domain that we are focusing on. It could be various aspects of healthcare. There's a big thrust from the government for AI in healthcare and so on, and lots of medical data, lots of genomic data on which we can apply AI and ML. So we need to be cognizant of all these challenges when we work on them. So that's like my broad pitch, and I'm very happy to take a few questions.
That ICMR already has a guideline on responsible AI, so which actually covers many of the things. So I'll probably share the document with you, and you can share it amongst others.
Yeah, I think Ravi was probably involved in drafting. Ravi heads this center we have on responsible AI, and I think he was involved in drafting of that. But my point was more towards actually getting research groups that are in this medical informatics and say AI imaging data analysis and so on, to actually look at many of these problems, right? So that's still underappreciated, and but definitely, I'll be happy to take a look at that.
So the idea is that in future, when ICMR or any funding agencies are going to dish out money, they would expect you to conform to these guidelines.
Yeah, I think everybody has to now submit a blurb on responsibility along with the proposals.
Yes.
Thank you.
Hi, thanks again for this lovely session so far, and thanks for the opportunity. So
So mine, I'll probably pick up from where Dr. Chandra left, because I feel like, that's kind of what I think is a grand challenge. In some way, I think it's like not necessarily practicable, but in other ways, I also feel it's inevitable that this is how healthcare will be administered, in the future. So what I try to show here, let's start with the blue side of this slide, and that's firmly the type of area that I come from. How do we decide how to cure diseases? So if we think of something like diabetes, we have 10 to 12 drugs in the market today. All of them, we figured out how to use that, how to administer those drugs based on clinical trial data, follow them up using epidemiological data based on some basic understanding of physiology and biochemistry. That level of solution really attempts at scale. In fact, we probably leave behind drugs which will help 10% of the population or 20% of the population, because drug development needs to happen in a way that, where most of the patients in the clinical trial are rescued, and that's kind of that scale mass solution, one size fits all, which comes from the right. And this is not just drugs, right? If somebody is trying a new diet protocol, if somebody is trying a new device, if somebody is trying any sort of new intervention, that how does that work at scale is how we, make those decisions. On the right-hand side is, where does this thing get personal? How do we tailor these solutions to us? And to be, today these decisions are, the personalization is very weak at best. So the one thing is what a lot of people in this room work on, which is kind of your God-given capabilities in terms of your genomic data. What does that indicate to you? How does that play out in terms of risks and liabilities in your life? Then comes, how does your own medical history and life history affect those? What kind of job do you have? What kind of life do you have? What kind of food have you been able to intake? Where have you been able to live, etc. The third one, which I think we just started touching upon a little while back, is, there's going to be a new deluge of data coming in from people having apps where they're able to, track themselves. And right now it's very cottage industry, but almost everybody has a fitness app. We all know, how to track our weight, etc. But soon we will have devices. Today there's continuous glucose monitoring devices. So these devices are approved for type 1 diabetics. So these are not one in 3 months measurements, these are continuous glucose measurements. It's not inconceivable that we think of a simple device which measures not just glucose, but maybe a panel of biomarkers yet to be decided. Maybe that will include cell-free DNA. What do we want to measure to monitor the health of a person? So, today, when you go to the doctor, most of it is what comes from the right. If you have a good doctor, then maybe your family physician knows your medical and life history and will tailor it a little bit for you. But really, when you go to the doctor, it is a one-size-fits-all solution. You can conceive of a future where, we are able to create databases where all of this information goes in, and, we are able to make decisions using AI, and this is where maybe Karthik's point starts to come in, where we start to crunch all this information and we start making recommendations, which are both leverage the best of what's available in scale solutions and tailored personally. So when you come, there's obviously a lot of thinking to do, when it comes to what kinds of solutions should such tools offer with the simple Hippocratic oath of do no harm. There are community health, goals, and we saw this a lot in COVID, right? Should I get a vaccine? Who am I getting a vaccine for? Should I take a mask? Am I taking a mask from what could be used for a healthcare provider? So there are community health goals which also have to be sort of factored in as we make these decisions. But last but not least is personal health goals, right? So again, I feel like the one-size-fits-all is something that everybody sort of chafes against. In the 80s, if you had to listen to music, it's pretty much what the radio plays, and whatever the Chitrahar guy decides to show, that's basically your musical taste. You only have access to that information. So I guess the millennials will have no idea what I'm talking about, because today if you go to Spotify, you can have a very curated experience just based on what you want from the same utility that we all share, and that can have be tailored very specifically to you. And what's more personal than health, right? So people can have many different choices they want to make with health. I may want a long life. Somebody else may want to live, fast and furious. They want to try everything, they want to eat everything, they want to drink everything. And those are reasonable choices. when you get diagnosed with diabetes, today when you go to the doctor, you're just going to get metformin and some general advice and an understanding that, hey, sooner or later you're going to get on insulin, and that's basically it. But on the other hand, if you factor all of these personal goals in, maybe you have the luxury to have lifestyle changes that you can incorporate. Maybe you try lifestyle changes for 6 months and you're like, this is not worth it, I want to get back on metformin. All of these are personal health choices, which today the way we administer healthcare is not really being factored in. And I think the negative aspect of that is, people push against it. They go towards unscientific and sometimes unscrupulous, administrators of healthcare, and kind of, that's where the trust in the system is lost a little bit. So, this is how I think healthcare will be administered for our grandchildren and great-grandchildren. But, let's see how far we can get with that. So, thank you for the opportunity.
I just have one question that increasing such a large amount of data, and that too a personalized data, wouldn't that increase the risk of security issues, like because it will have a data with a name on it, with
For
a
sure.
specified person and everything linked to it.
Yeah.
So what can be done regarding that?
I don't know enough about data security, but that is a really important point. If we do have to, draw good walls around it. On the one hand, I feel that. On the other hand, I feel like people readily give away their data. If you look at any Apple Watch, any Fitbit, people are very happy to throw their data around. So I'm not saying that's correct, but I'm saying, we need to have a solution that's both practical and sort of respects people's, people don't mind sharing data. We just have to kind of find a way to use that cleverly.
Who are doing. So that's going to become much more challenging when, let's say, a corporate is going to ask you to run 5 miles a day just to make sure that besides your mind, they can control your body as well. Sorry to the corporate here. But the other thing that I have a problem with is, how do you define harm?
Yeah.
Because when you are personalizing an experience and you are saying that I want to live fast and die young, is that your doctor's recommendation?
Yeah, may not be. So I don't know how to define.
And how do you define somebody as unscrupulous, for that matter, right? Because maybe it's a social construct of long life, right, or a healthy life, but maybe in a different society, a different kind of a life could be a more fulfilling life, right? So this is, I think, when you talk about personalizing a health goal, whether if it's talking about losing 5 kilograms, right, versus delaying the onset of diabetes, or I think that has to be looked into a little bit more finesse as to what kind of health goals you are proposing in this kind of model, and what can you actually predict on a single person's lifeline or timeline based on what you have already observed from the collected data.
So let's take, and to be clear, right, I'm really a modeler in drug development. So this was a 5-minute talk, so this is kind of out of syllabus, but it's okay. I'll try my best to sort of think about some of these things. So, what about data privacy and who gets to see this data? I think these are questions not only in the scope of health, retail, finance, everywhere where we start collecting data and make big decisions. I feel as communities we need to start thinking about how to do that. And maybe some of the things that what Karthik was raising, it'll have to be kind of a joint decision. It's not one person's thinking, obviously. The next question that you asked about do no harm, right? That's an old adage, and in that context, it's very personal, right? Don't lop off a lesion which may kill a person, so sort of in a very limited context like that. But and I think all the way to AI systems, that adage is kind of blanketly used, and I've used it in that sense. How to define harm, that's the hard work that we'll have to do. When it comes to sort of these moral ethical decisions, right, you're right. I think what it is has to be defined by many different people. But what I feel is, when you work in health, like all of us do, at the end of the day, biology which impacts health, I think there's no getting away from the fact that everything that you do, any recommendations that you give, there is an ethical angle at the end of it that, you know, we have to participate in, not necessarily as experts, but as one kind of voice on the table, because whether we like it or not, all of the work that we do eventually rolls over to affecting somebody's life. So I think whether we like it or not, those decisions have to be made. I think you asked one more question. Yeah, how do you define who's unscrupulous? You're right, there is a spectrum. You know, if you have some doctor who holds your hand and talks to you, you know, how much is that placebo effect worth? And, you know, that is an important thing to say. I'm not saying that anybody who doesn't follow PubMed is not being useful to the patient. I'm not saying that at all. But I think on the other hand, there are straight up unscrupulous people who, you know, take advantage of patients' vulnerabilities to kind of hawk all sorts of stuff. So there is a spectrum there. I'm not saying anybody who treats a patient with good intent is necessary, but unscientifically is necessarily unscrupulous.
I have tried to, and given that we have about, well, an hour, if people are ready to wait, like stay on for a little longer. We are planning for 2 to 4, but it's 3:20 already. If everybody is fine with staying till 4:20, then maybe we can do an hour session of discussion. Okay, I'm not hearing any problems anywhere, so let's. 4:15. Okay. And the other thing is. The other thing is that I have tried to divide this into 2 themes. Hopefully those themes make sense to you guys. If not, let me know. And we can try to change it around a little bit. But then, so that we get at least half an hour per theme, and we can have a panel discussion theme-wise. So 2 half-an-hour blocks of 1 theme each, and try to come up with something. So it seemed to me that there are 2 kinds of proposals here. One is a bottom-up thing where we are either trying to create some data or like collect data or build technology. So we had discussed about atlases, we discussed about combining atlases, we discussed about modeling. So these are technologies which are not specifically tied to one particular end goal, but can be applied to multiple end goals. So this is a bottom-up approach. And as far as I understood, Dr. Ishan, Debarka, Ramesh and Manikandan, they fit into that theme. And then there's another theme which is sort of top-down. And here I've taken a little bit of liberty. So responsible healthcare, it seems to me that if it's tied to a top-down goal, it makes more sense, or it is easier to drive. So those kind of things I've done here. But so it's targeted towards a high-level goal. We can maybe pick a target disease or some target problem, and then build technologies for that. And here I think that Professor Nagasuma, Karthik, Rukmini and Ramesh, again, they fit into this. I'll quickly go over my stuff so that we have time for discussions. So as far as I saw in this case, in case of building bottom up, we have an approach which is similar to UK Biobank. We are developing a resource, whether it's data or technologies. So we can take a similar kind of an approach. And the next one, so okay, let me not talk about this one. In the other theme where we are doing top down, it's sort of similar to cancer moonshot, right? So we can, let's say, pick type 2 diabetes or a rare disease, and then go to through the top down process. So this is how we think that we can try to sort of control the discussion. So in the half-an-hour block, maybe we can start with an exploration hat. And I would like to invite maybe the panelists of the first theme while I'm talking about this, Professor Ishan, Debarka, Ramesh and Manikandan. So the idea is that first 15 minutes we just discuss in an exploratory fashion. People are just trying to understand. So if there are any questions from the audience or any questions across panelists, we can do that so that we understand the thing better. Then the next 10 minutes, we try to have a consolidation hat where we try to come up with a sort of a more compact proposal. And I'm not fooling myself. I don't think we are going to get a really compact proposal out of this. But the idea is to sort of get people thinking and get people to talk to each other. Maybe we can, once we go back, we have an idea of who we want to talk to and continue this discussion further. And then in the end, we'll have a summary session. So we'll do the same thing for both the sessions. Let's start with the first theme. And just to confirm, the themes make sense to people, right? Anybody has any? Okay. Audience questions? Any other odd questions among the panelists? There is, yes. But maybe we can share. There are 3 mics for 4 people. That should be enough. So I can.
Maybe I can start. So I think Ramesh mentioned that I think this circles back to what Sabri was asking. Are these samples amenable to such, like blood samples, amenable to sequencing, or you have permissions for doing so?
Yes, so these things have all come in for sequencing. They got sequenced using whatever, I think a couple of years ago we were doing smaller panels. Now, then it moved to whole exomes. So they've been sequenced using something, and they do have, invariably they will have research access. In most cases it's easy enough to, in situations like this, go back to an ethics committee and say, these are anonymous samples being used for research, do a waiver of consent or something like that, because they've been sitting around for a while. So I think the ethical and regulatory consideration is relatively light. We can get through that. It's the scientific and figuring out how to solve the problem that's the bigger challenge. Was this in this theme or second theme? How are you?
I think the rare diseases is in the second theme.
Okay, yeah, I guess as much. But so that leads to a good question on, maybe I can lead, since you're talking about the regulatory issues of how do we, if we collect samples, how do we get the right permissions? Of course, if you go through a process of collecting, writing up a project, getting ethics approval for that project, then of course everything is fine. But well, the only challenge is if you have to do it at a scale where it's collaborative amongst multiple parties and there's funding from sources abroad, etc. Then there's a fair amount of red tape that it goes through. You might have experienced it. And
is it possible? So I think collecting tissues throughout the body might be a challenge, but if we can start with blood, which is already there, if this blood is preserved, flash frozen, ideally RNA shouldn't have degraded. One can isolate nuclei and.
So for the rare disease problem, you're saying, or for all these
CFDNA,
the other ones? Yeah.
For whatever you have samples available.
Oh, so what we have samples available for the rare disease problem, we would have gotten blood and we would have banked it at whatever minus. What do we bank it at? Minus 80. So that would have been done. So whatever state it is in, I think whatever one can do with blood banked at minus 80 for it's been collected over the last few years. So that we can do, certainly. From the larger perspective of all of these projects, for example, cell-free DNA, it doesn't help because when you just bank blood, whole blood, the cells break and then they dilute the so-called cell-free DNA. That's DNA outside the cells. So the big challenge with cell-free DNA is that you have to collect in such a way that you can store it by removing the cells out and store only what's called the plasma, the liquid part of the blood. In that plasma, you will not have any cells, so the cellular DNA will not dilute the cell-free DNA. The cell-free DNA is coming from all the other cells in the body, not necessarily from, a large chunk of it will come from the blood cells as well, but at least you have a hope that there's enough that comes from all the other parts of the body. If the blood cells start breaking, then they'll dilute that cell-free DNA so much that the signal as it is, which is weak, just goes to nothing. So the challenge we have with doing cell-free DNA atlases using existing blood banks is that most existing blood banks or existing blood storage processes would not have taken into account the fact that you have to separate the cells from the plasma almost very close to the time of collection. It has to be done like within a few hours, or if you collect it in special tubes, you can do it within like 24 to 48 hours. So that's the challenge why all cell-free DNA work pretty much has to be done from scratch, all collections.
So I have a question on the cell-free DNA. For detecting cancer or whether you have a particular gene or not, we do blood samples also. Is this the same thing, the cell-free DNA, or is that a so suppose
it's a different thing in the sense that you take a blood sample and it has a lot of things in it. So you've got the red blood cells, you've got the white blood cells, you've got all kinds of platelets and other stuff that clots, and then you have the liquid part that's left over called plasma, which has a range of molecules in it, very few cells, but a whole lot of molecules in it. Those molecules are cell-free DNA molecules that are coming from various cells in the body. They are metabolites, proteins, all kinds of things are sitting there. And that is the part that you look at, because that gives you a window into cells across the body. Now, if you do a typical blood test like an LDL, etc., you don't need to take the plasma out. You do it from the serum. Serum is a place where you get all these other molecules also, but you don't get hold of the cell-free DNA from there. Right? So I think there are different fractions in the blood that have to be carefully looked at for different sorts of tests, and some things work in certain fractions. So it's not all the same.
Okay. So I follow-up question. I had to get this done for someone recently, unfortunately, and what I was told by the sequencing company that there are fragments of DNA which get into the blood from the solid tumor, and those are the ones which we measure. Is it
correct?
Similar
That's
to this?
right.
Okay.
Yeah. Okay. So fragments of DNA will come from all over the body. Hopefully some of them will come from the tumor cells, and you can figure out they're coming from the tumor cells. Well, they don't have an ID on them, but so you can figure out they're coming from the tumor cells because they carry certain hallmarks. Those could be mutational hallmarks that typically if you have a mutation in a certain gene, sort of there's high likelihood that it's a certain sorts of mutations are high likelihood being from a cancer cell, let's say. There are epigenetic hallmarks, certain methylation patterns and things like that. So while there is no ID on a fragment saying I'm coming from a tumor cell, these hallmarks you can look for, both genetic and epigenetic hallmarks, and ascribe them reasonably to saying there is a tumor lurking underneath, whose properties I can get from looking at these hallmarks. Right?
Okay. One more question, unless anybody else wants to.
Yeah. So in tackling some of these problems, you need like an entire ecosystem to revolve around it, right? For example, you need trained human resources. You need like some sort of supply chain that's able to handle the requirements of this sampling, storage, and things like that. Maybe infrastructure in certain cases needs an upgrade. So I think his point on the slide is very nice. That do we have all that is required in India, and what would be the gaps that we need to first focus on?
It's a good question. I can take a shot and then pass it along. I think all of this is eminently possible in India. Now let's look at what all do we need. We start with the ethical framework. We need to, well, firstly, even before that, we need to have a bunch of what are the groups that need to come together to solve this problem, right? There will be molecular biology groups, people who understand molecular biology, people who understand the clinical world somewhat, saying what is the clinical implication of all of this. Eventually blood is going to come from patients, so therefore the clinical folks will be the gatekeepers of recruiting people in hospitals or outside. And so you need the clinical world, you need the molecular world, you will need people who of course the engineers and the assay designers and the data analyzers and the data modelers and all of that, right? And then you'll need to go through various regulatory processes, so one will need that expertise. All of those are available in plenty all around when I see it. I mean, there's no dearth of any of those today. There are also a number of programs going on where people run these cohort studies, They, I was just across the road at the Center for Brain Research. They are running 2 cohort studies here in Bangalore. There's a Tata longitudinal study that's running of 1,000 patients. They're recruiting 1,000 normal individuals and trying to follow them up over a period of time to study neurodegeneration and things like that. So a number of these studies are going on, so it's nothing new. So what we will need to do is to, one, get some of these groups together, get a vision in place saying this is the sample collection we want to do, this is how we're going to store the sample, this is what we are going to assay the sample for, and these are the potential endpoints that we can get at the end of this process. Getting it through the ethics committees and all are relatively easy, as long as if there's international funding involved, takes a little longer, there's more complex processes, otherwise it's relatively easy. And then if you have clinician collaborators, they will be at the hospital sites, recruiting them, recruiting patients, drawing blood, all of that they'll do. Then the blood has to come into some blood bank. So we'll need a facility where you have some freezers. Many labs have the ability to, most labs have the ability to do some amount of storage. If one needs larger than that, let's say tens of thousands of samples or something, then one can, it's just a question of getting enough freezers and space and having the protocols in place. There are enough and more biobank protocols that one can borrow from worldwide to get best practices and all of that. There's careful attention needed to storage so that you don't waste these samples. So once one does that, then designing whatever assays one wants to run, looking at sequencing, or there might be other things, metabolomics, etc., whatever, there's a whole range of things. So we'll need experts in all of those areas to participate. And then it's back to this group, which is perhaps the data modeling and analysis and all of that. I think we have everything. In terms of expertise in the country, in terms of volunteers, patients, samples, logistics and infrastructure to collect samples, bring them to a central place, freezers to store. None of that is a fundamental challenge. Around the room, if you ask, there will be somebody or the other who's done all of, some piece of this or the other, and it'll together add up to make the whole. Pass it along.
So I would just add. Yeah, I would just agree with that. We have all the pieces. So specifically for the modeling part, for instance, we can use some of the data that are being generated to learn or tune some of the parameters rather than learning from the literature, right? So it's the main challenge will be to come up with a set of people who can rally around a common goal. So that I believe is, there are examples that these cohorts are being generated, but there are some challenges or the other even on these ongoing cohorts in terms of data sharing and data access, data modeling across different groups. And this is not specific to India. All across the globe, there are consortiums which have these issues, but they work it out. And so we need to get these people together, interested people, and that I believe will be a bigger challenge rather than kind of different aspects which we already have.
I have actually a question on this. It seems that from actually as representing the Strand here, a lot of such initiatives with much blue sky expectations are being carried out in the West by startup companies, not even established companies. How are they able to fund these things? And do you think that as an established outfit, is there possibility of doing this outside the need for government funding, even crowdsourcing such an endeavor?
Yeah, this is a very fundamental question. In fact, if I were to also connect it to what you were asking, I think we have all the expertise, all of the infrastructure, all of that is not a problem. What we need is the ability to sell a vision and get funding for it. Because these things are not, while they'll be a lot more inexpensive here than in the West, they still are a substantial expense. So one has to raise money, and to raise money, one will have to paint a picture of the impact that this can have in terms of unlocking further research, in terms of making a difference to the lives of patients or whatever. One will have to be very clear about that and rally around on that theme. So it will not be anymore each of us solving our individual research problems. We'll have to sort of see that big theme and line up against that, and then canvas for money. Now, coming back to your question, is what is the best way to raise money for this? So in the West, obviously the West always has more disposable money to invest in lots of things, and therefore you see larger quantums of investment. But you're right that what I'm seeing over the last several years is there's a lot greater interest even in the private sector to invest in several of these things. And often I see that right at the back of some infrastructure that's been built. For example, UK Biobank decided 15 years ago, 20 years ago to just collect samples. Once those samples were collected, a whole bunch of private investment came in saying we will do all the MRIs, we'll pay for the MRIs, we'll pay for the sequencing, we'll pay for this, that, etc. Once there is some established infrastructure, I think getting funders to pay for things becomes much easier. With the Indian government, I think we'll have to follow a multi-pronged strategy. I think the government, of course, is there, and we should figure out how to come together and get a vision together and write grants to the government. In parallel, I think if we can paint the vision well enough, we can go to the private sector is a lot more open now than it was. But again, you don't see the levels of investment with the private sector unless they see the path very clearly. Right? But smaller amounts of investment will be possible, I think. And then the onus is on us to get the trajectory in place so that with those small amounts of investment, we can get the path clearer and paint the picture much more clearly so that from the private sector, we can generate larger amounts of investments. So speaking of Strand for itself, right, today there are a few areas where we wouldn't mind paying for the sequencing. Right? For example, if there's somebody has a cohort of 1,000 people with a certain disease, then and the clinical data around it is well characterized, then that's something we've reached a stage where we're at least open to that, right? Which was not the case with companies like us several years ago. So those sorts of, so I think we are at the cusp of, it is still not a slam dunk, it's still not just go to one place and get funding, we'll have to start. But if we take this as a 5-year process, I think India has evolved enough that if we start today, we will find multiple sources of funding from the private, from government, from larger companies, etc. But we'll have to paint that vision well of what the impact of all of this can be. And if we can also paint the vision in such a way that what comes out of this is valuable to all of these companies in some form, then it becomes even more easy.
So I would like to add two-pronged thoughts here to this whole discussion. So number one is when it comes to the academia, because I have also burnt my fingers or have been partially successful in bringing technologies into a commercial diaspora, but it has never been very easy, right? So we, for example, you were talking about this ctDNA based or cell-free DNA based methods for diagnostics. So we have done it with tumor-regulated platelets, which are more available, because the one problem with cfDNA based approaches is that if you are looking for certain canonical mutations, then you might not find it all the time, but whenever you find it, it will be very good validation, and so the specificity will be very, very high. The sensitivity will probably not be as high, but there are different modalities. For example, GRAIL is looking at methylation patterns instead of mutations or any other thing. So it is fairly successful, I would still say. But what I really look for as an academician, primarily academician, is that we have a lot more hand-holding when it comes to marketizing or commercializing our products. There's still all these incubation centers are there. So I come from IIIT Delhi. So we have an incubator, we have everything, but even then they don't have the expertise. Nobody understands the regulatory process. Where is the step 1, step 2, step 3? some extent validating it on small sample sizes where you are very confident is fairly easy. But after that, it is not only the money problem. People are interested to give money, as you just mentioned. I know a lot of people who are ready to invest to the tune of a few crores, which is a good seed money for an Indian startup. But the problem comes when nobody knows, even in that company, that how to take it forward through the process of UNESCO approval, this approval, that approval, and so on and so forth. I think that is what I would say we need to talk more about, like how to really activate those commercial avenues from academic research, which really have commercial potential. Number two is, I personally think, because so ignorance is bliss, and I really didn't study genetics very well. I'm a computer scientist. So when I looked at this genetics and Jiva's studies, I felt that these are still using these age-old methods, and drawing statistical power essentially primarily from the sample size alone. And that to me is a very top-down approach. So you look at a lot of observations from that, whatever is your enrichment of a particular phenotype or so, you are probing deeper into it, so you are going by observation. And modeling guys are doing something with a few parameters, a few variables, and few genes, so they are doing it in a different silo, right? Genomics guys are doing in a different way, whatever. Now the point is, now we have these large language models. I actually wanted to talk to you about the mutation embedding work that we have done, which does not really use the power of sample size. Instead, it learns from the flanking region context, right, as a language model. And that can detect even a pathogenic variant, even if it is appearing for a single time. So that's a very preliminary, rudimentary method, but I think this has great potential. So we really need to understand how the pathogenicity arises and what are the epistatic effects, pleiotropy, and all of that, in a very different way. We should not be too much reliant on statistical genetics approaches that are old good methods. So what is your or anybody else's view on that? I'd be very happy to. You have probably done the most work in this area.
So on both of your first point is, how to commercialize. It's a bigger conversation, I guess, beyond the theme of this. But where it connects to the theme is only to say, how will we, if we put an umbrella like this together and try to raise money from multiple constituencies, how will we make it appealing to the private sector, right? So that we can get participation. I think it's a good question. We'll have to figure that out.
It's also about hand-holding the post-invention processes.
Hand-holding, yeah. So that's what I mentioned is the much larger. It's a whole topic for different discussion, and we'll get there perhaps another time. On your second point, one of the big challenges in the bio field is lots of things work on small data sets and get tumor-educated platelets, you mentioned, work beautifully in 1 or 2 papers, then never got replicated. A few things sustain the test of time, and those get replicated in multiple labs, even if the person who's doing the experiment is not the most qualified, it still works in spite of that. The sequencer changes, it works in spite of that. Lot numbers change in the reagent, it works in spite of that. It works in spite of the ambient temperature. You need a lot of robustness before something can be commercialized. And the attrition levels are so high that it's pretty much going to be, even so in the last example you gave, the pathogenic, everything will work on some whatever data set you have. You now open it up to whatever millions of, now we have the whole UK biobank, etc., right? And you open it up, you'll see a fair amount of misses on all sides. And so the onus of taking this all the way to the end has to be on the originator, and they have to somehow say, okay, I've now got on a 100,000 size data set, I've got decent predictions. Where do I get hold of a 500,000 size data set, how do I show this is robust? How do I show that it is making some systematic errors which I can eliminate, etc. Until such point that it becomes ready for prime time, right? And then the commercial process starts. I think that's after that there are all the risks of return on investment, commercial competition, logistics, this, that. I mean, hundreds of things come after that.
Alluding to that, the actual question that I had was, do you feel after doing all this for all these long years successfully that the existing statistical genetics methodologies are somewhat inept and can be upgraded?
The general question, you
like typically just only relying on the sample size, even if you just sample the whole human race, will you be able to understand the genotype-phenotype relations?
So the way, good question. The way will we be able to understand? I mean, today again we'll be digressing, but briefly since you mentioned this, yesterday I was watching the printer running. It was spewing out printout papers. Each paper would come out, float, and land at a different place on the floor. And I said, let me predict where the next page will land. I was horrible at it. There is no hope. I mean, you cannot predict where that next page will land. So complex systems suffer from the problem that small changes in input lead to a large change on the output, and there's no way, unless you can measure, you can't measure with all the variables with. So I don't think we'll be able to, even with all the data on Earth, be able to predict everything, because these are the complex nature of the beast is too much. However, there'll be enough utility we can get out of this that can make a real difference to life, health, healthcare, disease, and all of that. Right? So that's the point. And the point to your whether statistical genetics, I think we should just, problems are hard enough that we have to just pull every single trick that we have. If it is more data generation, be it so, if it's statistical genetics, if it's modeling, is it whatever it is. I think we have to dig deep, because none of these problems will get solved very easily.
So I'll have to interrupt. We are severely out of time, and in the to be fair to the next round of panelists, we'll have to end.
Have a ending on how do we culminate and take this forward?
Yeah. So I mean, I just wanted to say one thing. It seems like I'm hearing from everybody that we have the required expertise, and technology, whatever is required, is available in India. Funding is the main thing, and it seems from Human Genome Project or from UK Biobank Project that the funding is in the domain of the government right now, because private companies will be interested after the data is, or after there is some data generated and there is some utility shown. So we should be going towards the government for funding.
Are we planning to?
We can. After this we can try to follow up and see where it leads to.
That would be good outcome, I guess.
What is the anticipated scale of the project?
Those are matters of detail. We'll have to.
Yeah, let's do it in the next.
Anticipated, right? I'm not saying. So X in X years or something.
110,000. And X dollars.
So the goal should
100 genomes in 1,000 years.
I think we should aim for anything that's, it has to be at a certain minimum scale. I think so. We should aim for like 100,000 type scale.
Yeah.
Now, we will not get there in one shot for sure. We'll have to start with 1,000. We'll have to continue selling the vision, stay on a theme, and continue, and then try to see how we can build this up to something bigger. But,
Okay. Yeah, thank you, panelists. Dr. Ramesh, if you can stay on.
I'll quickly start off with one important challenge, because I think Professor Kumar talked about deep phenotyping and so on. I think the first sort of problem, also partly related to what he was mentioning, is that there's just so much variability. Right? And what is a healthy person versus what is an unhealthy person, or because if you look at any aspect, and I would like to think about it from the microbiome perspective, there's just so much variation. And similarly, even if you were looking at the genetic parameters or any other parameter, there's just so much variation where a healthy person stops and where an unhealthy person begins. is such a challenge, and this is a big confounding factor in any kind of phenotyping you want to do. So I think this is one big challenge that we need to address as we look at these large-scale phenotyping and genotype-phenotype models and so on. How do you handle this plasticity?
Okay, it's not as hard as we imagine, because we can define ranges based on well-known clinical phenotypes. Right? Defining health is difficult, but it's not that difficult. If we understand that it is a wide range, it is not likely to be, for example, take blood glucose. We will say 80 to 100 fasting sugar is normal, something of that kind. If it goes to 79 or 101, we might have some sort of a penalty for that, but not too much. So how we actually capture that data, I think, is very important. We still need to define some ranges, cutoffs, otherwise we will not be able to proceed. So likewise, I think, is somebody healthy or not, we could measure things like simple neutrophil lymphocyte ratio will tell us something. Things like CRP, blood is a very, very good tissue for that. So you can measure a wide range of things which are actually available in the clinic today, and many people are doing that. Measure, taking the example of diabetes, glucose, HbA1c, LDL, triglycerides, HDL, all of these things are very important things to tell us what is healthy, what is not. So it should be, feasible.
I think a little bit where that starts to become important is when do you intervene, right? So, do you intervene when you're at 110? Do you intervene when you're at 120? And those, again, I think there are well understood kind of long-term studies and simulations about where that should go, but I feel like that again may be a little bit problematic. The other thing is, I think there are some diseases like diabetes which are really good because literally it's defined as hyperglycemia and the biomarker is also glucose. So that connection between disease and biomarker is very sharp. But if you think of even autoimmune diseases, right? CRP is a weak measure. It can go up and down if you have arthritis or you don't have arthritis, for IBD or not IBD. And not even to mention much more complex diseases like mental illness, right? Like if you could think of depression or things like that, there's literally no biomarker. So I think I kind of agree with both sides. There are some cases where that delineation will be easy, and there'll be some cases where it'll be hard. And in all cases, I think you look to clinical practice and clinical trials to figure out when to intervene. Yeah. Okay. So the point I was trying to make was for research purpose. For the discovery purpose, then we'll be very stringent, define a range. Your point is really important when it actually comes to practice later on. So then you can open up and then say they don't have to be binarized into healthy and disease. It's actually a continuous spectrum. So quantitative kind of interpretation becomes important.
So the other question I had was, if suppose we are targeting, let's say, type 2 diabetes, or sorry, I forgot. No, not cancer, but the rare diseases. Yes, rare diseases. What is the scale of data which we need to collect for that? Is it similar for both,
I
or?
was just doing some mental maths as the first panel was meeting. I think if we aim for about 3,000 patients, the cost that one can even apply for, hope to apply for, right?
So suppose cost is not a constraint.
Then I would like to have 10,000 patients at least, right? Cost is not a constraint, but that is a constraint, so we have to be realistic. So suppose you are looking for funding in the range of say 10 to 15 crores. The maximum you can cover is about 3,000 patients, around that, with all deep phenotyping and everything associated with that. This is just a very, very rough estimate.
But I'm trying to keep cost out for the reason that instead of thinking in this order that, okay, this is the amount of money we have, this is what we can do, what is it required to solve the problem? If we want to understand diabetes thoroughly and want to solve it, then what is it required?
I don't think there's a very strict defined limit. So, 1,000 to 3,000 would give us plenty of data.
Okay.
So there are consortia in Europe which are doing that currently. I don't know how many samples are being taken, but something in this order.
Okay.
I think that would give us enough population level statistics power, which will give us confidence to go back to individuals as well, personalization.
So I think maybe the one, sorry. So, one of the things is, how to solve the problem. I think it'll actually help to define what exactly this problem is, right? So if we're talking about early diagnosis of diabetes, that's a different scale problem. You need both normals and pre-diabetics, and then watch how many, actually unfurl into diabetes. So early detection is one sort of problem. Personalized treatment. Like I said, today there are about 10, 12 drugs on the market, including lifestyle interventions that you can give to a patient. What is the first drug that you want to give, and how would you give it? So there, I think both the genomic type data and all of the blood measures as well as clinical history would be really useful to kind of then make assessments on what is the best treatment for that person going forward. Question is very important.
Yeah. On the rare disease front, it's very simple. Like you can do, as I said, we have I'm sure if we dig in, we'll have a few thousand undiagnosed cases. We pick even 100 from them and throw the kitchen sink, and we are able to solve out of those 100 another 10 or 20. You've already made progress, right? And so the whole.
Can you elaborate a little bit on what you mean by solve? Is it like finding out what is the genetic change which leads to the phenotypic symptoms?
Convincingly pointing, pinning down the genetic change as associated as being the cause of the disease.
So then would you say then the next convincing would be like a knockout model or something like, or like, a treatment with the missing protein or take off the excess protein, like what is the bar for convincing?
Yeah, so if one is able to pin down something in a gene which is known to be the cause of that phenotype, then that's all. Then that would be, and you're able to show that that gene is severely impacted in some form, then you perhaps don't need anything more. If it's a mutation in a gene that's known to be involved in the phenotype, but the mutation's effect you don't know, then you'll need to do some functional work. If it is a completely new gene, etc., then one would have to associate it with the disease in some form and study. So there'll be a few different things to be done, but 100 will give more than enough fodder to pin down, like even if we look at those 100 and say do some initial sequencing of various kinds and come down to maybe even 10 that look most promising, and then we do a lot of study around those and say, okay, we are able to solve 10. It went through a long, complicated process, but now I can systematize it into something simpler that we can now run repeatedly.
So that's 100 per disease, right? Per rare disease.
No, this 100 is
okay.
Because there is no. I mean, the disease range is enormous, and it's
right.
Yeah, just 100 would be enough work and enough impact if we.
So let me ask the question in reverse order, and maybe I'm muddling the two sessions then. But UK Biobank, they collected data for 500,000 individuals, and they're doing more, and they are of course working towards personalized medicine or personalized healthcare, which is sort of the theme of this particular panel. So what kind of problems are there which would require that scale of data to be collected, and what additional things can we learn from them?
Yeah, so I mean, there is applications at the personal level. One person has a problem you're trying to diagnose, and that answer lies only in that person's DNA or epigenome or somewhere inside that person. There are other problems that are sort of more complex disease conditions, multifactorial, age-related. No one individual you'll be able to pin down anything from there, so you need much larger data sets to say, if I have to pin down this particular location, the DNA as having some role, and its role is that it raises your risk by 20%, or you just have to have large data sets to be able to pin it down. So those are the two ends of the spectrum. The rare diseases end is the easiest end, and that's why people have looked the hardest at it, because it's individual, it is 1 or 2 mutations that cause the problem, and pinning it down at an individual level. We are able to do in 50-60% of the people in a clinical delivery setting. Sample comes in 2 weeks, report goes out, stuff like that, right? That's like extreme industrialization. So that's it's gotten there, except for we can only solve 60% of the case. Complex diseases is just very open.
Right. So suppose we were to, let's say, create a proposal for the government where we want to collect data at a very large scale. What would be the top level, if we are doing top down, what will be the top level problem that could drive something like that? Because bigger data will lead to more technologies getting innovated and all that. So I'm trying to focus more towards the larger scale. So how do we sort of motivate that?
One thing that occurs to my mind readily is the genetic basis for diabetes. Why are Indians more genetically oriented towards getting diabetes? That's an important question, both fundamental question as well as it will have translation potential. That can be solved. And maybe early diagnosis, and which of them can be reversed with lifestyle changes, and what sort of lifestyle. So these are, like for example, simple breathing, what we know as pranayama, whatever that may be, that how much can be reversed. So these are all individual questions. We can't bunch them up into one proposal because we have to collect data very, very differently for each of these things.
Right. It may not be one proposal, but it can be one program.
One
right?
program.
One program from the government which has multiple research projects under that umbrella.
Exactly.
Okay.
So I have maybe a question on that. Sorry, I came. With all these sort of technologies which are said, at the end of the day, the intervention which is said is live a healthy life, don't have intoxicants in excess, get regular checkups. Is there really a intervention which is not obvious that is expected to come out of such a longitudinal?
No, with the best of lifestyles, some people are still going to get diabetes. With the best of lifestyles and the best of circumstances, they will still need interventions to control their diabetes, right? So there is a spectrum there. Today, whatever you do, you go to the clinic, you're going to get an intervention for diabetes. But then there is a spectrum, and that's where maybe this deep phenotyping stuff can help, right? That is, if you look at 1,000 people with diabetes, do they cluster in ways that is phenotypically different and hence interventionally susceptible to different interventions? I don't know that we know the answer to that. And like I said, right now the solution to a disease like that is one size fits all. You come in, doctor will advise lifestyle changes, then metformin, then sulfonylurea, then SGLT2, and then insulin. It's just, you're on that track the whole time. And that probably is necessary for 30% of the patients, but probably not for 100%.
So there's no evidence to say how much each of that actually contributes, and that's what we are trying to get at a deep level. We don't have real scientific evidence, which is why many people are not convinced. They say everybody says, "Okay, do exercise, and why should I do it?"
And there is growing evidence on the genetic aspect as well, right? I think Mohan's Diabetes Center, they've accumulated a ton of data on this count, and they see that this maturity onset diabetes of the young and so on. There are clear genetic markers, and you could have the completely wrong treatment if you didn't run a simple genetics panel on it, right? So I think there is a mounting evidence in terms of this. So time to actually go deep and have money.
No, I was going to add that simply defining the cutoffs, right? So even that can come out of this
kind of a study where is 125 fasting glucose, is that the limit? I mean, currently it is based on certain evidences from Western studies, but in our Indian populations, will it be the correct? Is 6.5 the correct? Yeah, is 6.5 the correct for our setup? And also to partly address your question also, I was going to add to the comments that we have to probably start with one sample as a pilot and then figure out what the required sample size is for the later ones, because there are just too many unknowns, and we won't know until we have a first look at the data.
Right. So even to form a proposal for the government, we probably need to do a pilot study.
Pilot study, yeah.
Multiple of us need to come together and do that.
Yeah.
Right.
Some pilot studies are ongoing. Like, for example, that we have something going on.
Right.
Others will also
Right.
could use that as material.
Right. we are talking amongst ourselves. Any questions from the audience?
Yeah, that's sorry.
Very nice session. This is more a generic thing. On one side, we will have more understanding of the data variance and everything. How do the, or like, what is the anticipation that the people will understand this over a time? Because we also have a debate about removing evolution and other things from the textbooks and so on, and it's kind of keeping it for the higher classes. So how do you see collectively all these things that people will prepare to accept all these things? Sorry for the broader thing, connecting many things, but just to...
I just don't know to answer it.
See, I think we are generally very open to technology, open to everything, right? So this, in fact, during this AI and so on, right? India is apparently one country where people think AI is acceptable, right? And whereas you see in Europe and all that AI is highly feared, and it will take away jobs and so on. So I mean, as a society, we are very open to everything. It seems like, and so that seems to be something that may work in our advantage, may not work in our advantage. Yeah, there are so many other issues that we need to deal with, lots of education at different levels and so on. But looks like this is something that people will be, oh yeah, let's try it out, kind of thing.
We are very aspirational.
Yeah.
Okay, what I was going to say is, if we are able to translate that knowledge into some actionable items, such as there is a test, a person walks into your clinic and their entire family is advised, if they have that genetic basis for it, to be more proactive in changing some lifestyle, or just making them aware of what sort of food or what sort, it need not be pharmacological all the time, right? Simple things of that sort. I think that will go a long way. I think lack of awareness itself is a problem, and to give awareness you need scientific evidence and data, and that's what this will create, is my opinion.
Okay, so we are sort of getting out of time. Are there any parting thoughts before we wrap up?
Get us funding from somewhere. We are all so starved these days.
I think, do the youngsters have anything to add?
Just one question. So,
Yes.
yeah, like we have all this databases for intra-species database, that is human species database. But what we can learn from comparing interspecies, like we do all the time model, our model is not a human but an animal. So we also need to know the difference between disease manifestation, evolution between two different organisms, how much we can extrapolate and how much we can understand from that, what kind of knowledge that can bring to this kind of research.
That's more fundamental, right? I mean, it is important to understand evolution of, say, insulin signaling pathway, or how things have evolved and how regulation has happened. But those are different questions. Whether that will feed directly into clinical aspects, it's a long shot. So that will be a little more, longer route, basically. So while it's interesting and important, it should be for its own sake. We want to learn about the species and evolution for its own sake, which is important. I don't know if we can mix the two easily and put them together, which would be harder to sell. Yeah.
Okay, let me conclude. Sorry, we are out of time. From what I understood was that if we can sort of find a high-level driving application which can create a program, and of course we may have to start with a pilot thing, or we can rely on pilots which are already going on. But if we can find a high-level thing which can tie multiple things together, whether it's responsible AI, whether it's AI for healthcare, whether it's different kinds of diseases, or collecting data and all that, having that all together will make a difference, I think. And if we have been struggling to get funding or struggling to make a case for funding, then maybe we'll need to get better at marketing our idea, right? And one criteria, from what I understand, is that we have to make that person look good who is giving us funding. So if Modi can get on a podium and say that, oh, I'm going to do this, and that can win him elections, that's when it will happen. Otherwise, that kind of large-scale program will not happen. So we'll follow up. Let's maybe continue the discussion and see if we can
Maybe
take.
this one last question from Mr.
Sure.
So I hear that a lot of translational suggestion from the doctor is lifestyle-based changes. But what's the motivation to have drug development for a precise group of subgroup? You're basically reducing a market for a drug development. So where does the interventional pharmacology come in the picture? For drug development or the interventional clinical trial where you are doing the subtyping and then giving? Because you mentioned one statement where 20% of the people...
Yeah, that's almost an economic and a question of incentives, right? So it's true, most of my day I spent on trying to make better drugs, but me and my colleagues in kind of a much maligned industry for several good reasons, I think every new drug that we make is useful for some people. Is it useful or necessary for all people is a different question, right? And what other changes can happen in the environment to even prevent such diseases, right? So even in the US, where a lot of this happens, I think just changes in the food availability, fresh food availability in an area, that itself can reduce incidence of metabolic diseases, things like that. So back to your question, I think society is in general made of several entities with their own incentives. But when we talk about an effort like this, where kind of the objective function is all of society, not necessarily one part of society. As a drug developer, I will do everything to come up with a better drug, but as a citizen of the society, I would like kind of an optimal solution for everybody versus something that just optimizes one part of society. I hope that...
But then the precision therapy is a lost cause if you want to, like we're starting the research with the thing that we want to treat every subgroup and every subgroup has a different source of the cause of the disease. But when it comes to translation, we are also saying that we can't develop a drug specific to that subgroup.
I don't know if we necessarily say we can't develop a drug. Today the incentives are so aligned that a scale solution is better than a precise solution. But that doesn't mean that's the globally optimal solution.
Yeah, and there is a lot of examples of very, very specific solutions also. I mean, I'll give you one example. This drug for cancer treatment, if you have a mutation in this gene called NTRK, NTRK123, very extremely rare mutation. If you sequence the 1,000...
For the illuminating discussion.
Yeah. Hello. Yeah. So thank you all. I think this concludes the scientific part of the symposium. So great talks over 2 days, great question answers. So it was very exciting. We are very happy and satisfied with the outcome of this symposium. So I want to congratulate all the poster presenters yesterday for putting their energy and putting a lot of energy in presenting their ongoing research. I wish them all the best that they convert their ongoing research into good publications. So we had several faculty members who reviewed the posters as well, and we requested them to identify a few outstanding posters so that it serves as an incentive for you to also come again next year and again submit posters. Right. So may I request Professor Ramesh Hariharan to give best poster awards. So the 3 best poster winners are Dia Binay Joseph. So she presented the poster Identification of Spatially Restricted Mesenchymal Factors in the Developing Lower Urinary Tract. Is she here? Nihar Lakhe for presenting poster Comparative Genomics of Epigenetic Components to Understand the Evolution of Parasitic Behavior in Protists. and Shreyansh Srivastava for presenting drug discovery in time, studying binding pocket dynamics of Rev ERB. Can we give a big hand to all the poster presenters? So I just want to thank everyone who contributed to the success of the symposium. So on behalf of my colleagues, Professor Devnath Pal and Sanchit Mishra, I want to thank all the speakers. So we were grateful to have exciting lineup of talks spread out throughout 2 days. And the questions that were asked after the talks, that were pretty exciting. So I think many of the questions that we got most of the questions we got from students. So that was pretty exciting. And so besides speakers, I want to thank our student volunteers. So please. So I think they worked tirelessly for last 4 days. So I finally will get some sleep now, right? And so our secretarial staff from Axis Bank Center as well as CDS and SCRC. So as you know, success of this symposiums like this depends on them. So they handle the logistics of your accommodation, travel, and so on. So a big round of applause for them as well. Finally, our helper staff, Shivana and so please Shivana and Manoj. And Ramu, yes, thank you for taking care of all the activities and keep the symposium running. And finally, I want to thank our sponsors, Axis Bank Center of Mathematics and Computing. So we were able to run the symposium without charging any registration fee. So we were able to cover all the meals, all the venue costs. So I want to also thank them for covering the cost of the symposium. And hopefully we'll get more money from them to run the symposium again. So, and I'll see you again next year. Thank you. So, final remark is that we'll be circulating a feedback form to you all. So, if you have any feedback for us on how we can improve next year, please do. So, that feedback form will go to you before 6 p.m. today. So, please let us know if you have any feedback. So, anything I missed, Professor Devnath Pal?
Yeah, if you give good feedback, then we have a meeting again. Because this is a request from Axis Bank that we get feedbacks because they know that, this is a philanthropic CSR funding that has been given to us, and they are very thrilled to see full house. And I think if it's backed up by good feedback, then guaranteed we will be next year.
Right.
So high tea.
Yes, so now we'll break for high tea, and that concludes. Thank you all.